So calcium is a very unique mineral. It's not only a nutrient, but also a signal. When it serves as a signal, it actually can regulate the homeostasis and use efficiency of other mineral nutrients. So this is a topic we're going to discuss today. It's going up and down the arrow that doesn't seem to work.
Yeah, it works.
Yeah.
Oh,
Look
okay.
at these. Oh, it's fine, I think.
If you want this, you have the laser pointer also.
Oh,
You
okay.
can see,
Okay.
so I
Ah,
go
the
back to
pointer
that
here.
one, yeah.
Oh, okay. This is good. Yeah. Okay, so plant growth need four bare necessities, including light, CO2, water, and mineral. So in natural setting, light and CO2 are plenty. Actually, CO2 levels rising, we know that. But water and mineral nutrient are often limiting factors for plant growth. So as a result, crop production or agriculture, we use a lot of irrigation and fertilizers. These are the two major input for modern agriculture. But this is not very sustainable. Taking fertilizer example is contribute heavily to the yield increase. About 50% of yield increase in this half of the century actually should be attributable to fertilizer increase. If we look at this chart, this number is quite dramatic. This blue is a population increase, and the red is fertilizer use. By the middle of this century, we're talking about 9 billion people, and the annual use of fertilizer is about almost 300 million tons, right? So this, you see these two curves go hand in hand, and the fertilizer use is really not sustainable. Not only some of the fertilizers are natural resource and you use it up, like petroleum, you use it up, it's gone. But also, of course, this fertilizer use and agriculture practice contribute to this vicious cycle, right? So we know that climate change has a lot of negative impact on agriculture. Agriculture, on the other hand, produce a lot of greenhouse gas and that contribute to climate change. And it's well known that fertilizer runoff cause eutrophication of fresh water resources, and then really damage our ecosystem. So sustainable agriculture is so important for the society, environment, and economy, for our well-being, basically. And we need to have a new revolution to support sustainable agriculture. And in terms of the fertilizer use, we need to increase the nutrient use efficiency to reduce the use of fertilizer. So nutrient use efficiency depends on many processes, including root uptake of mineral from soil, transport from root to other organs, and redistribution of nutrient throughout the developmental processes. And for some perennial plant or crops, the recycling of the mineral nutrients at the end of the growth cycle is very important. So all these processes are limited by membrane transport. There are a lot of transporters mediate that process at a single cell level. The membrane transport not only control the in and out of nutrient but also the distribution of nutrient within the cell in different organelles. So ideally, the nutrient homeostasis is really for all the organelles in a cell and all the cells in a plant get a perfect balance of nutrients. This is something that each plant is trying every minute to achieve. So how does plant cell do that? Basically, there are two networks. One is a signaling network that detect the signals inside the cell or demand signal. So how active is the metabolism determine how much nutrient you need inside. And of course, they also need to detect how much is available or the supply. Then the signaling process can decide. We look at this one. So there are really two big networks, right? One signaling network that detect a supply and a demand, and then inform the transport network that consists literally hundreds, thousands of transporters that kind of inform the transport network which transporters need to work, which transporters need to be inactive. So it's a very dynamic process. So my lab has been trying to understand the integration of these two networks. And within the signaling network, calcium is a very important signal messenger, not only in plant, but also in other eukaryotic systems. Right, in response to different external or developmental cues, plant cells often raise the level of calcium in the cell, and that serves as a signal messenger. That actually transmit different signals into different physiological responses. So the big question mark here is really how does calcium, a simple canine, translate so many different signals into distinct physiological responses. What I mean is really, plants respond to high temperature and pathogen differently, right? But in the middle, calcium is always there. So where's the specificity derive? So the research in the past about half century also in both plant and animals, especially in animal systems, invented or defined a new term called calcium signature. That is to say that different signals produce different calcium signatures, right? So this calcium signature is determined not only by the concentration in this case, the peak, height of the peak, but also the spatial and temporal patterns, right? It's truly four-dimensional parameter. For example, we can clearly tell the difference between the blue pattern and the orange pattern, and cells can tell that too, and they interpret different patterns by a process called the decoding mechanism or decoding steps. And that often start with the calcium sensors that bind the calcium and then change their own conformation and activate downstream players involved in gene expression, for example, transport process. And then the whole cell will calculate all these changes and give a specific output. We call it specific response. So from specific signals to specific response that entail coding and decoding of calcium signals, right? So in my lab, we've been working on both the coding and decoding mechanisms in the past 25 years or so. And I'm going to give a couple examples quickly on the coding mechanisms. So how are those patterns are produced at a molecular level? So one of the example is pattern triggered immunity and plant in response to a bacterial pathogen. We know 30 years ago from Dirk Schill's lab, actually, he found that calcium was an important messenger for plant pathogen response. But the mechanism how calcium get into the cell has remained unresolved until a few years ago. We find a CNGC type channel that actually play an important role in this process that mediate the pathogen-induced calcium entry. So without pathogen, and this channel is inactive, everything is kind of not uncolored, it's inactive. And in response to pathogen patterns, the pattern receptors get activated, and they in turn activate downstream protein kinases such as BIG1 that phosphorylate one of the subunits of the channel and then turn on the channel. So without pathogen, the channel is closed because chemodulin binding. After phosphorylation, somehow the channel is open. The mechanism of opening of the channel still remain at the structural level, still remain unresolved. This is one of the channels that are that are involved in pattern response, pattern triggered immunity. Sorry. And as you probably realize, in the past 5 years, the calcium become a central theme in plant immunity, right? Some of the ETI or effector triggered process involve their effector receptors. Some of the receptors are calcium channels themselves, right? So both the CC type and the helper Rs, they form calcium channels. So calcium it really become a central hub in plant immune response. Another example is a plant reproduction. So that begins with pollen grains landing on the stigma, and then the pollen tube delivers the sperms to the ovule. Each step of the way, from germination to pollen tube guidance, growth and guidance, to reception or birth and fertilization, involve calcium. See, each step there always you see calcium, and this calcium is featured with oscillation. And I'll show you a real picture here. So this is a pollen grain. Before germination, you already see the oscillation of the calcium here. And then upon germination, it follows the tip of the pollen tube. It's also flashing. Flashing means oscillate, right? So we can look at this one more time. So flashing, go up and down like this pattern. The germination, follow the tip of the pollen tube. Flash, flash, flash. Yes, so, and this process involves the calcium oscillation at a molecular level. We just published a few years ago that it's involved 3 molecules, the 2 CNGC subunit plus chemodulin under low calcium when the cell calcium is low, and at the bottom here, chemodulin is empty, and it actually act as a regulator by associated with this inactive channel, and then active the channel. So the channel open and the calcium enter plant cell, and the cellular calcium will go up, right? When it go up, it binds the chemodulin, and the chemodulin loaded with calcium will dissociate from the channel and shut, then the channel will be off again, right? So in this case, and of course it will start a new cycle. In this case, calcium serves as its own pacemaker, right? So it's an auto-regulatory feedback mechanism, right? So this is in the context of an earlier talk on the pH oscillation. I thought it's a kind of interesting to relate. So when the pollen tube reaches the ovule, right? So there's a gatekeeper cell, we call it synergy cell, would actually sense the raw peptide release from pollen tube and oscillate, produce a calcium oscillation. Now this oscillation is at a molecular level is due to the binding of ROF with pheronia and LRE receptors. And then that co-receptor recruit a third component, NTA, which is actually a calcium channel. And then the calcium get into the cell. When the calcium reaches a certain level, it binds chemodulin. In this case, chemodulin bind to the channel and shut the channel down, basically close the channel. So you will have up and down, up and down. This is, again, an auto-regulatory process. Calcium serves as its own pacemaker. Yeah, I'll show you a video here. Yeah, this is a oscillation, calcium oscillation in response to ROF peptide in the synergy cell. Right. So, okay, so the last example is a systemic signaling mediated by calcium. This process is basically, this example is a wounding response. Okay, it's a severe wounding. You have to really cut the vasculature before the local signal get transmitted to the distal organ, become a systemic signaling process. So this process involves several signals. One is an electric signal, just like action potential in our nerve, really. And then calcium wave, we call it calcium signal, calcium wave, and then ROS wave. And all these three are required for jasmonic burst at the distal organ. Jasmonic acid, of course, the defense hormone, trigger the wounding response. Okay, so some 10 years ago, at Farmer's Lab, actually first shown that in terms of electric signal, it required two glutamate receptors. Those are the glutamate receptor 3.3 and 3.6. And the glutamate receptor, of course, is an important players in our neurotransmission, right? So a long distance or systemic signaling in our system. So I'll show you how this two, actually, we use root to shoot signaling as a system, as a model, right? So what we did is to cut the root. You have to really cut it, and not small minor wounding doesn't work. You have to really cut the vasculature. And then within half minute, the whole rosette get light up by the calcium sensor. This is a calcium reporter, right? So if you look at it, we can also use glutamine to elicit this process. At the wounding site, you can actually apply some glutamine. Then it's also mimic the wounding response. So we, of course, also report that both glutamate receptors from plant are also glutamate-gated calcium channel, and that are desensitized by chemodulin. Again, it's an auto-regulatory process, right? So you see it's go up and down very quickly, right? So within a couple of minutes, it's already gone. 30 seconds, 1 minute is pretty much done. So this up and down, the down curve is actually inhibition or desensitization by chemodulin binding to the C-terminus. Okay, so that's all, a few examples to show you how these patterns are generated at the molecular level and in the different contexts, right? So next, I want to introduce a couple examples of how the decoding works, and by talking about CBL, calcineurin B-like protein. So a while ago, when I was a postdoc, well, more than 30 years ago, it turned out now. It's amazing. Okay, so I worked on this animal molecule called calcineurin. This is a calcium-dependent molecular switch consisting of 3 subunits. Calmodulin and calcineurin B subunit are both calcium sensors or calcium binding proteins. Calcineurin A is a protein phosphatase. In response to elevated calcium, these 2 bind calcium form this trimeric complex activated protein phosphatase. So when I started my own lab, we began to look for similar molecule in plant, and we never find this kind of molecule, but we find a calcineurin B-like protein, calcium binding protein or calcium sensor. Now, how does this sensor work? Is to find its target, right? In this case, it's a protein phosphatase. In this case, it's a protein kinase. It's a SNF-related kinase or SNRKs, right? So SNRK1 type, SNRK2 type, 3 type. And this belongs to CIP, we call it CBL interacting protein kinase or CIPK. This belongs to SNRK3. So it's a big family of protein kinases. So in a way, it's represents some kind of a paradigm shift, right? From animal fungi, they all use the calcineurin, the protein phosphatase, to respond to calcium or decode calcium, right? And of course, there are other mechanisms as well. So in the case of plant, we have a calcineurin B-like molecule, CBL, interact with a bunch of protein kinases instead of protein phosphatases. Biochemically, it's the opposite, right? Phosphorylation versus dephosphorylation. In plant, this is a big network, actually. In Arabidopsis, very small genome, right? So you have 10 CBL, at least 26 CIPKs. They interact with each other. Not everyone, every CBL interact with each CIPK, but there's some selective interaction. Some CBLs share common CIPK target, and some CIPK share common regulatory subunit. So there's both specificity and overlap in this biochemical network. Now, in the past 25 years since this was discovered, a number of labs trying to put this biochemical network into a functional network. How plant use this CBL-CIPK network to decode calcium, right? So I'll give you a couple of examples that's related to nutrient use efficiency. After all, we have to get to that point, right? The title, get to the title subject. So I want to use the potassium nutrient as an example. Okay, we know that potassium in the soil, it's usually at a micromolar or sub-millimolar level, quite low. And the root hairs use a couple transporters to get the potassium in and then delivered all the way to the xylem and then go up to the, let's say, mesophyll cell, right? Use this one as a sink, cell. So here, the cells will keep a lot of potassium in the cytoplasm and store excessive potassium into the vacuole, right? So there are 2 mechanisms plant use to deal with the low potassium in the soil. One is to boost the activity of these 2 doors, let the door wide open, okay, activate these channels and transporter to let more potassium in, right? So that's one. And the second mechanism is to get the stored potassium out of vacuole, right? Just like what we do actually for weekly shopping, right? So we can get more to our storage or into the refrigerator, or we can also retrieve our food from the storage. Okay, so, but all the doors and the refrigerators are locked usually. Okay, so you need to open those doors in order to get the potassium either into or out of the vacuole, right? Those two mechanisms actually are both controlled by the CBL-CIPK network. I'm not going to the 20-year work, right? So just to show you that the summary, what I mean by the calcium CBL-CIPK pathway regulating the low potassium response in plant. With the low potassium, there's a activation of the calcium channel. We still don't know which channel is involved in this one. Hopefully we'll find it soon. And then the calcium would be sensed by CBL19 and recruiting CIPK23 to the plasma membrane where they activate the uptake transporter, okay, get more potassium in. In parallel, another pathway, CBL2 and 3 and 4 CIPKs get recruited to the vascular membrane. There, they actually phosphorylate and activate TPK, which is the storage door that is locked. This is the key to unlock the door to get the potassium, store the potassium into the cytoplasm. As you can tell that both pathways, they have the same goal, that is to increase the cytosolic potassium to support the metabolism and the cell growth. So through the years, a number of labs find that CBL-CIPK network actually is really control nutrient use efficiency pretty much, or homeostasis. So there are many transporters, nitrate, ammonia, that's nitrogen, right? Phosphate, not yet. Potassium, I just told you, both plasma membrane and the vascular membrane or potassium transporter controlled by that. And then there are magnesium transporters, there are iron, there are almost all manganese. You know, most of the minerals are transport and utilization are controlled by CBL-CIPK network. Okay, so as I said at the very beginning, calcium is a mineral nutrient by itself. Does calcium as a signal control its own use efficiency or its own homeostasis? The answer is yes. Oh, I do. Okay.
You have still a bit of time. How long?
Oh, okay, so a couple of minutes.
A couple of minutes.
Yeah. So I want to use this. I hope this video plays. It's undergrad students made a video that actually show us, how this two pathways plant use to regulate calcium homeostasis. Do we hear? Oh, let me explain. Okay, so when the plant have a low calcium level, so the channel is not active, and then the vascular transporter that sequesters calcium also not active. So low calcium level, no problem. You don't need any of those mechanisms, right? But most of the time, normal soil contains high level of calcium, and that would leak into the plant cell through the channels, right? And I showed you earlier, it's a CNGC2 and 4 that channels. So, and then when it reaches certain level, it activates CBL-CIPK, and that in turn activate a sequestering calcium transporter, get the excessive calcium into the vacuole and reduce the level of the calcium. Calcium is toxic, by the way. Okay, so again, this is an auto-regulation, right? So when calcium level reaches certain level, it activates CBL-CIPK, get rid of it, right? In response to pathogen patterns I showed you earlier, it activates this channel, and then not synchronized. Synchronized. Yes, now, okay, phosphorate the channel, get the channel potassium, calcium in, and set the signal, but it need to remove it, right? So it's transient. And this same kinase actually phosphorylate this sequestration transporter, get the calcium into the vacuole to reduce the, going up and down. So one kinase have a dual function is set the signal and then reduce it, reset the signal. Okay, does that make sense? Yeah, so that's in summary, two different pathways. The calcium dependent CBL-CIPK pathway actually respond to high level of calcium to always keep the calcium level low, right? In response to patterns, the PRRs activate the kinase. The kinase on one hand actually set the calcium channel by activating the calcium signal by activating this channel, and subsequently it activates the vacuolar transporter to remove it, to set this transient peak, right? So, yes, I think I'll stop here. And hopefully, yeah, my title works, right? So the calcium signal